and welcome to Introduction to Biology, Bio 105. In this lecture, we will discuss photosynthesis. We're going to be looking at the light reaction and the Calvin cycle, which is also called the light-independent reactions of photosynthesis, and see why they're important in the process of photosynthesis. So, if you can remember back last few lessons, we discussed the structure of the leaf. If we zoom into that leaf, in the middle layer, in the mesophyll, are a bunch of chloroplasts. Actually, hundreds of chloroplasts can usually be found. And if we look at just one of those chloroplasts, we can see that there are different structures to help photosynthesis occur. And these structures are really unique to chloroplasts and its functions. And if you can remember, inside the chloroplast, which is made out of an outer membrane and an inner membrane, are these pancake-like structures called thylakoids. And these thylakoids are stacked on top of another, just like pancakes, to make granum. And the grana form a third membrane layer. The fluid-filled layer, aqueous fluid, is called the stroma. And this is going to be very important because these structures, especially the thylakoid membrane, which is the membrane of the disc-like pancake structure you see in here, is involved in the light reactions, whereas the stroma is involved in the Calvin cycle. The stroma has the enzymes that make the Calvin cycle work. Now, if we look even closer at the thylakoid membrane, so this is the membrane of one of those pancake-like structures, you see that there's two areas, the thylakoid membrane and the thylakoid space. So you have two photosystems in the thylakoid membrane, photosystem 1 and photosystem 2. Remember that this is because of the way they were discovered, not the way that they work. It's actually photosystem 2 that is the first one, and photosystem 1 is the second one. And what happens is when light hits the photosystem 2, it energizes the electrons in the pigment center so that they become excited and will then go through the cytochrome complex electron transport chain, which then will pump protons into the lumen or the thylakoid space, creating a high amount of hydrogen ions in the thylakoid space concentration. And the way the electrons in the photosystem 2 here that are excited and go down this electron transport are regenerated is by splitting of the water molecule. So the hydrogen ions also get pumped back into the lumen, increasing the hydrogen ion positive charges in the thylakoid space, and the oxygen gets released, whereas the 4 electrons found in these 2 water molecules are used to replenish the pigment centers of photosystem 2. So light will hit also the photosystem 1 and excite the electrons that are found in photosystem 1 to reduce NADP to NADPH. And then what happens is these protons that are sitting in the thylakoid space will be utilized to flow back through ATP synthetase to make ATP. And this ATP and NADPH will then be used for the second part of photosynthesis, which is the Calvin cycle, to make carbohydrates. So how do light reactions generate ATP and NADPH? Well, it's a similar mechanism to what we found in cellular respiration, in which you have a bunch of proteins on the membrane that serve as electron transport chain and pump hydrogen ions across the membrane to create a hydrogen ion gradient, a chemoosmotic gradient, which then the ATP synthetase will use to make ATP. Now, the main difference is in cellular respiration, food provides the energy for the electrons, whereas in photosynthesis, the light excited electrons will flow down the transport chain during photosynthesis. As I recall, as I told you, there's two photosystems, photosystem 2 and photosystem 1. The main difference is in photosystem 2, you have the electrons that are replenishing the electrons that are being flowing through the pigment center by water. The water is being split so that the protons stay in the thylakoid lumen. The electrons will replenish the pigment centers that get excited, and the oxygen is released into the air. In photosystem 1, the electrons that come to replenish come from the electron transport chain, and the chlorophyll A pair is replenished through that system. So if you look at this picture, here's the stroma of the chloroplast, here's the thylakoid lumen, and this is the thylakoid membrane. So you have photosystem 2 here, photosystem 1. Light is hitting photosystem 2, energizing the electrons so that the electrons will be carried through a series of protein carriers and proteins that pump hydrogen into the lumen of the thylakoid lumen, and this electron will then get passed on to photosystem 1, where it will replenish the electrons that are excited and lost to reducing NADPH. NADPH is reduced by NADPH reductase, and the hydrogen ion gradient that's found positively in the thylakoid lumen will flow back to the outside of the stromata by using ATPase synthase to make the ATP molecules needed for the Calvin cycle. So light reactions will harness the energy from the sun and the light energy to produce the chemical bonds of ATP and NADPH. These energy-carrying molecules are made in the stroma, where the carbon fixation will take place for the Calvin cycle. Now, the Calvin cycle is organized into three basic stages: fixation, reduction, and regeneration. In fixation, you have the addition of CO2 to this molecule or enzyme called ribulose 1,5-bisphosphate carboxylate, or Ribisco. And the three molecules of ribulose bisphosphate will then make 3-PGA, which is 3-phosphoglyceric acid. The PGA has 3 carbons and 1 phosphate molecule, and each turn of the cycle involves only 1 RUBP and 1 carbon dioxide and forms 2 molecules of the 3-PGA, or the 3-phosphoglyceric acid. The number of carbon atoms remains the same as the atoms move to form new bonds during the reactions. The process is called carbon fixation because CO2 is fixed from an inorganic form to an organic molecule. In stage 2, you have the reduction of 3-phosphoglyceric acid by the splitting of ATP to ADP and NADPH going to NADP. So the NADP and the NADPH, the ATP and the NADPH are used to convert the 6 molecules of 3-phosphoglyceric acid, or 3-PGA, into 6 molecules of a chemical called glyceraldehyde 3-phosphate, G3P, shown here. That is a reduction reaction because it involves the gaining of electrons by the 3-phosphoglyceric acid. Recall that reduction is the gaining of electrons of an atom of a molecule. And so the 6 molecules of both ATP and NADPH are used. For ATP, the energy is released with the loss of terminal phosphate atom, converting it to ADP. For NADPH, both energy and hydrogen atoms are lost, converting it to NADP positive. Both of these molecules return to a nearby light-dependent reaction to be reused and re-energized. Stage 3 is the regeneration of the RUBP molecule, or the ribulose bisphosphate molecule. Only one of the glyceraldehyde 3-phosphate molecule leaves the Calvin cycle and is sent to the cytoplasm to contribute to the formation of other compounds needed by the plant. Because the glyceraldehyde 3-phosphate exported from the chloroplast has 3 carbon atoms, it takes 3 turns of the carbon cycle to fix enough net carbon to export 1 glyceraldehyde 3-phosphate. But each turn takes 2 glyceraldehyde 3-phosphates. Thus 3 turns takes 6 glyceraldehyde 3-phosphates. 1 is exported while the remaining 5 glyceraldehyde 3-phosphate molecules remain in the cycle and are used to regenerate RUBP, which enables the system to prepare for more carbon dioxide to be fixed. 3 more molecules of ATP are used in this regeneration reaction. So in summary, the Calvin cycle will take the carbon dioxide found in atmosphere and make sugar out of it. It acts like a sugar factory within the chloroplast in the stroma. So it'll use the carbon dioxide, the energy from ATP in the light reactions, and the NADPH to make the molecule of the glyceraldehyde 3-phosphate, or the G3P molecule. So light-independent reactions occur in light or dark. Some enzymes require activation by light. The light-independent reactions, or the Calvin cycle, occurs in the stroma of chloroplasts, and they require the ATP and NADPH that was generated from the light reactions to reduce the CO2 and make carbohydrates and sugar. So CO2 is converted to sugar by entering the Calvin cycle. So the Calvin cycle takes the CO2 and makes it into first phosphoglyceric acid, and it uses this enzyme called ribulose bisphosphate carboxylase, which is then the most abundant protein on Earth and is regenerated back into the cycle after you have the molecule of glucose being made. So it does it through 3 stages where you have fixation of the carbon dioxide, reduction of the 3-PGA, and then regeneration of the ribisco molecule. Stage 2, you have these light-independent reactions. It does not require light, but occurs in the stroma of chloroplasts, requires CO2, and uses ATP and NADPH of the fuel to run it, and the end product is glucose sugar, or CO2 and